students, welcome back to the science class. Continuing our lesson about the nervous systems or coordinations, today you're going to learn about how the transmissions of nerve impulses, action potential, and membrane potential. So without further ado, let's begin our lesson. Transmissions of nerve impulses. Neurons transmit electrical impulses. These impulses travel very rapidly along the cell surface membrane from one end of the cell to the other and are not a flow of electrons like an electric current. The transmissions of nerve impulses is called action potential, caused by the very rapid movement of sodium ions and potassium ions into and out of the axon. Membrane potential. Membrane potential refers to the difference in electrical charge between the inside and the outside of the neuron. This is the plasma or cell membrane of the neuron. It separates the inside of the cell from the outside environment. We'll say this is the inside and this is the outside of the neuron. The difference in electrical charge develops due to the grouping of ions on the inside and outside of the membrane. Ions are atoms that have either lost or gained electrons and thus have a positive or negative charge. There are several ions that play an important role in the membrane potential of neurons. There are positively charged sodium ions represented by these blue circles and negatively charged chloride ions represented by these green circles. When a neuron is at rest, the sodium ions and chloride ions are more prevalent outside of the cell. There are also positively charged potassium ions represented by these yellow circles and various negatively charged ions, often referred to as organic anions, represented by these gray circles. Anion is simply a term for a negatively charged ion. When a neuron is at rest, the potassium ions and organic anions are more prevalent inside the cell. At rest, the inside of neuron is more negatively charged than the outside, causing the resting membrane potential of an average neuron to be around negative 70 millivolts. One way this potential is maintained is through a mechanism known as the sodium-potassium pump. This is a transport protein that uses energy to constantly pump 3 sodium ions out of the cell while at the same time pumping 2 potassium ions into the cell. Because there are more positive ions being pumped out than in, it helps to keep the membrane potential negative. Unlike other ions, potassium tends to move fairly easily across the cell membrane through ion channels, which is a membrane spanning proteins that allow ions to pass through. Potassium will pass out of the neuron until it reaches the point where it is at equilibrium, when forces like diffusion aren't pushing it in one direction or the other. At this point, the membrane potential of the neuron is around negative 65 to negative 70 millivolts, which is known as the resting membrane potential. Now, let's talk about action potential. Before we discuss more about action potential, there are a few terms that you need to know first. Depolarization refers to a sudden change in the membrane potential, usually from a relatively negative to positive internal charge. Repolarization refers to the change in the membrane potential that returns it to a negative value just after the polarization phase of an action potential, which has changed the membrane potential to a positive value. This is a neuron, which has 4 main parts. The dendrites receive information. The cell body processes and integrates that information. The axon carries the information along long distances from one part of the neuron to another, and the axon terminal transmit the information to the next cell in the chain. A bundle of axons traveling together is called a nerve. Nerves can be very long, as they often need to transmit information over long distances. As we just saw, The dendrites are the part of the neuron that receives incoming signals. Based on the strength of this incoming stimulation, the neuron must decide whether to pass this signal along or not. If the stimulation is strong enough, the signal is transmitted along the entire length of the axon and the phenomenon called an action potential. When this happens, we say the neuron fires. Transmissions of neuronal signals is entirely dependent on the movement of ions or charged particles. Various ions, including sodium, potassium, and chloride, are unequally distributed within the inside and the outside of the cell. The presence and movement of these ions is not only important when a neuron fires, but also at rest. To start, let's think about the positively charged sodium and potassium ions. When a neuron is not sending a signal, it is considered to be at rest. In a typical neuron in its resting state, the concentration of sodium ions is higher outside the cell than inside. The relative concentration of potassium ions is the opposite, with more ions inside the cell than outside. This ionic separation occurs right at the cell membrane and creates a chemical gradient across the membrane. Because ions are charged particles, we also need to consider their charge when thinking about their distribution across the membrane. At rest, there are more positively charged ions outside the cell relative to the inside. This creates a difference in charge across the membrane, which is called as electrical gradient. Together with the chemical gradient we already mentioned, we refer to this ionic imbalance as the electrochemical gradient. The difference in total charge inside and outside of the cell is called the membrane potential. At rest, when no signals are being transmitted, neuronal membrane has a resting potential of approximately minus 70 millivolts. This means then the inside of the cell is approximately 70 millivolts less positive than the outside. Both the chemical and electrical gradients we just discussed contribute to stabilizing this potential. While the inside of the cell has a net negative charge and the outside of the cell has a net positive charge, the charges line up at the membrane, and the bulk solution of either side is actually electrically neutral. The resting membrane potential is the point where the cell has achieved electrochemical equilibrium. This means that the concentration gradient and the electron gradient for each ion is equal and opposite. Ions cannot simply move across the membrane at will. Instead, they need a protein embedded in their membrane to facilitate their movement. Most ions cross the membrane through a structure called an ion channel. Ions move through channels by passive diffusion along their concentration gradient. Some ion channels are always open, but many require signals to tell them to open or close. For example, voltage-gated channels only open when the membrane potential reaches a certain value. On the other hand, ligand-gated ion channels are triggered to open when they are bound by a specific molecule. Mechanically gated ion channels open in response to physical forces such as changes in length or changes in pressure. Most ion channels are selectively permeable, meaning that they only allow one or a small subset of ions to pass through. Voltage-gated ion channels, for example, typically only allow a single ion to cross the membrane when they open. This means that we need separate channels for each ion. For example, voltage-gated sodium channels as well as voltage-gated potassium channels.
As ions move through a channel and cross from one side of the cell membrane to the other, they cause the membrane potential of the cell to move away from its resting potential. If the resulting change in membrane potential is small, we call this a gradient potential. Gradient potentials can vary in size, can be either positive or negative, are transient, and typically do not result from the opening of voltage-gated ion channels. When ion channels open, a gradient potential occurs. The neuron moves quickly to reset its membrane potential to resting values. This is accomplished primarily by the use of the sodium-potassium pumps, which uses the energy generated by ATP hydrolysis to actively transfer ions across the membrane against their concentration gradient. In other words, sodium is transported to the outside of the cell where its concentrations is higher, and potassium is transported back into the cell where its concentrations is higher. One cycle of this pump transports 3 sodium ions outside the cell and brings 2 potassium ions inside the cell. This unbalanced charge transfer contributes to the separation of charge across the membrane and also to the ionic concentration we see at rest, thus restoring the chemical and electrical gradients to their resting levels. Maintaining this ionic balance in neurons is so important that this process can account for 20% to 40% of the brain's total energy use. Only when the resting membrane potentials and ion distributions are maintained at precise levels will the neuron be poised and ready to fire an action potential. When the outside stimulation is large enough to bring the membrane potential in the neuron body up from minus 70 millivolts to the threshold voltage of minus 55 millivolts or higher, this triggers an action potential at the axon helix, which then travels down the axon. Voltage-gated sodium channels have 3 states: open, closed, and inactivated. At rest, the sodium channel is closed. Once the cell membrane reaches the threshold voltage, the channel changes to an open position and sodium rushes into the cell because the electrochemical gradient. As positive sodium ions enter the cell, the membrane potential becomes less negative and more positive as it reaches 0 millivolts. This is called depolarization. Eventually, the voltage gradient goes to 0 and beyond 0 up to a positive 30 millivolts. This is called an overshoot. As the membrane potential becomes positive, the sodium channel inactivation gets shut, making the channel inactivated. This stops the flow of the sodium ions into the cell. The change in the membrane potential also opens the voltage-gated potassium channels, though they open and close more slowly. Because of the potassium electrochemical gradient, potassium ions flow out of the cell, making it less positive and eventually negative. This process is called repolarization. Because the potassium channels are a little slow to close, for a brief period, the membrane potential is hyperpolarized. It's more negative than the resting potential. During hyperpolarization, the potassium channels close. Throughout all of this, the sodium-potassium pump is still working. The pump restores the chemical gradients by putting the sodium and potassium back in place, and the pump re-establishes the potential gradient by moving more sodium ions out than potassium ions in. This returns the membrane potential back to its resting potential. During repolarization, the inactivated sodium channels won't respond to any stimulus at all. During this time, the neuron is in its absolute refractory period, the period of time when a nerve cannot fire another action potential, no matter how strongly it's stimulated. The absolute refractory period prevents action potential from happening again too quickly and prevents action potential from traveling backwards along the axon. During hyperpolarization, the sodium channels are closed and the inactivation gets opened. There is no change in sodium flow, but now they could be opened again. This is called the relative refractory period because while the sodium channels could open, it will take a larger than usual stimulus to reach their hold because the cell is hyperpolarized due to the potassium still leaving the cell. The amplitude of the action potential for a particular neuron, that is, the maximum voltage in one neuron during an action potential, never changes. An action potential doesn't get bigger with a bigger stimulus. It's all or nothing. It either happens or it doesn't happen. What can change is the frequency of the action potential. A neuron might fire many more times per second in response to, say, an intense pain or less frequently in response to a gentle breeze. Some axons transmit action potential faster than others. One variable that increases conduction velocity is the presence of myelin sits around axons. Myelin speeds up transmission through a process called saltatory conduction, in which the action potential signals appear to jump along the part of axon covered by the seed. In the peripheral nervous systems, the seeds are formed from glial cells known as Schwann cells. There are small gaps within Schwann cells called the nodes of Ranvier. The action potential appears to jump from node to node, speeding the transmission. In the central nervous systems, the seeds are made by cells known as oligodendrocytes. To review, with no stimulus, the membrane is at its resting potential. A small stimulus causes a gradient potential, and a stimulus above threshold creates an action potential and the neuron fires.
I think that's all about our lesson today. Don't forget to do the exercise on my mother. Bye!